Why are sea urchins used to study embryonic development?
Sea urchins are a useful model system for studying many problems in early animal development. Historically, sea urchins were a key system in elucidating a variety of classic developmental problems, including the mechanisms of fertilization, egg activation, cleavage, gastrulation, and the regulation of differentiation in the early embryo. In addition, early studies of the molecular basis of early embryology were carried out in this system. Gametes can be obtained easily, sterility is not required. And the eggs and early embryos of many species are beautifully transparent. The early development of sea urchin embryos is also highly synchronous, that is, when a batch of eggs is fertilized, all of the resulting embryos typically develop on the same schedule, making possible biochemical and molecular studies of early embryos.
What is electrophoresis?
Electrophoresis is a technique used to separate biological molecules, such as nucleic acids, carbohydrates, and amino acids, based on their movement due to the influence 532 of a direct electric current in a buffered solution. Positively charged molecules move toward the negative electrode, while negatively charged molecules move toward the positive electrode.
What are the four groups of seedless, vascular plants?
The seedless, vascular plants include ferns of the genus Pterophyta, which is the largest group, the whisk ferns of the genus Xylophyta, the club mosses of the genus Lycophyta, and the horsetails of the genus Arthrophyta.
Is there a gay gene?
Homosexuality has a strong genetic component. One study found that among 56 pairs of monozygotic male twins, concordance simultaneous incidence for homosexuality was 52%, while it was only 22% for fraternal dizygotic twins and 11% for unrelated adopted brothers. This indicates that the closer two males are genetically, the more likely they are to have the same sexual orientation. Several studies have pointed to a likely location for male homosexuality traits at the tip of the X chromosome. It should be noted that not all males with the sequence identified themselves as homosexual, and so it seems likely that a variety of factors, environmental and genetic, may have roles in determining sexual orientation. So far, there are no correlate statistics for female homosexuality.
What is the difference between magnification and resolution?
Magnification, making smaller objects seem larger, is the measure of how much an object is enlarged. Resolution is the minimum distance that 2 points can be separated and still be seen as two distinct points.
What is the difference between primary growth and secondary growth?
Primary growth occurs in the tips of stems and roots in plants, thus increasing the length of the stems and roots. Secondary growth allows a plant to increase its diameter. The results of secondary growth form the division of a cylinder of cells around the plant's periphery.
What is a mutation?
A mutation is an alteration in the DNA sequence of a gene. Mutations are a source of variation to a population, but they can have detrimental effects in that they may cause diseases and disorders. One example of a disease caused by a mutation is sickle cell disease, in which there is a change in the amino acid sequence. Valine is substituted for glutamic acid. Of two of the four polypeptide chains that make up the oxygen-carrying protein known as hemoglobin.
What is molecular evolution?
The study of molecular evolution is the study of how proteins and nucleic acids change with time. Early studies on evolution were based on biogeography and the fossil record. As scientists developed techniques for studying DNA and proteins, evolutionary researchers soon followed their lead. Molecular evolution involves two broad areas: how the forces at work on individuals, e.g., natural selection, sexual selection, affect gene sequences and proteins, and how entire genomes evolve.
What do the numbers on a bag of fertilizer indicate?
The three numbers, such as 15-20-15, refer to the percentages by weight of macronutrients found in the fertilizer. The first number stands for nitrogen, the second for phosphorus, and the third for potassium. In order to determine the actual amount of each element in the fertilizer, multiply the percentage by the fertilizer's total weight in pounds. For example, in a 50-pound bag of 15-20-15, there are 7.5 pounds of nitrogen, 10 pounds of phosphorus, and 7.5 pounds of potassium. The remaining pounds are filler.
How does caffeine affect cells?
Caffeine is probably the most common drug ingested by people worldwide. Caffeine affects cells by stimulating lipid metabolism and slowing the use of glycogen as an energy source. As a whole, the body responds to caffeine by extending endurance, allowing you to stay awake for longer periods of time or perform extra activities. Adverse effects of excess caffeine intake include stomach upset, headaches, irritability, and diarrhea.
What are truffles and where do they come from?
Truffles, a delight of gourmets, are arguably the most prized edible fungi. Found mainly in Western Europe. They grow near the roots of trees, particularly oak, but also chestnut, hazel, and beech, in open woodlands. Unlike typical mushrooms, truffles develop 3 to 12 in 7.6 to 30.5 centimeters, underground, making them difficult to find. Truffle hunters use dogs and pigs that have been specially trained to find the flavorful morsels. Both animals have a keen sense of smell and are attracted to the strong, nut-like aroma of truffles. In fact, trained pigs are able to pick up the scent of a truffle from 20 feet 6.1 meters away. After catching a whiff of a truffle scent, the animals rush to the origin of the aroma and quickly root out the precious prize. Once the truffle is found, the truffle hunter, referred to in French as a trufficulteur, carefully scrapes back the earth to reveal the fungus. Truffles should not be touched by human skin, as doing so can cause the fungus to rot.
What elements are common to all types of microscopes?
Three elements are needed to form an image: a source of illumination, a specimen to be examined, and a system of lenses that focuses the illumination on the specimen and forms the image.
What factors might influence the interpretation of DNA fingerprinting?
Ruling out procedural sources of error, such as contamination from another sample or improper sample preparation. DNA obtained from siblings will share both alleles at a particular locus, and so the DNA profile may not be conclusive. DNA from identical twins will have identical DNA profiles. The frequency of identical twins in a population is 1 in every 250 births. In all cases, the probability calculations that are used in DNA profiling are based on a large population. Therefore, if a population is small or reflects interbreeding, probabilities must be adjusted.
In what year was the European starling, Sturnus vulgans wingspan, imported into the United States?
Eugene Schieffelin, 1826 to 1906, imported the European starling into the United States in 1890. Schieffelin wanted to establish in the United States every bird found in Shakespeare's works. He also imported English sparrows to New York City in 1860.
How does the mass number of an element differ from the atomic number?
The mass number is the sum of the number of protons and neutrons in the nucleus of an element. Using helium as an example, the mass number of helium is 4. As it has 2 protons and 2 neutrons in its nucleus. Since it has only 2 protons, the atomic number of helium is 2. When the atomic number changes, i.e., the number of protons changes, the result is a different element.
Why is RNA an unstable molecule?
Not all forms of RNA are unstable. Messenger RNA, mRNA, molecules, however, can vary in their stability. Depending on their rate of degradation and synthesis in cells and the amount of a particular protein needed.
How could a human be cloned?
Nuclear transplantation or somatic cell nuclear transfer is used to move the cell nucleus and its genetic material from one cell to another. Somatic cell nuclear transfer may be used to make tissue that is genetically compatible with that of the recipient and could be used in the treatment of specific disease. Or, if the material is moved to an egg cell lacking its own nucleus, the transfer could result in the formation of a clone embryo.
What are functional groups?
There are numerous patterns of atoms and bonds that are found frequently in organic compounds. These configurations of atoms are called functional groups, as each has specific, predictable properties. For example, an amino acid contains both an amino and a carboxyl, while alcohols all have a hydroxyl group. This group results in the solubility of alcohol in water.
What disease is caused by protists of the genus Plasmodium?
The sporozoan Plasmodium causes malaria. An individual of the genus Plasmodium enters the human body through a bite of a mosquito of the genus Anopheles that has been infected by the protist.
What molecules are most important to living organisms?
Four molecules are referred to as bioorganic because they are essential to living organisms and contain carbon. Nucleic acids, proteins, carbohydrates, and lipids. These molecules are all large, and they are formed by a specific type of smaller molecule, known as a monomer.
What is a karyotype?
A karyotype is a snapshot of the genome. A karyotype can be used to detect extra or missing chromosomes. Chromosomal rearrangements, or chromosomal breaks. Any cell that contains a nucleus can be used to make a karyotype. However, white blood cells seem to work best for human karyotypes. After the cells are cultured, they are killed by using a drug that halts mitosis. And the chromosomes are then stained, observed, and a size order chart is produced.
What are the major groups of echinoderms?
There are 6 principal groups of echinoderms, from the Greek terms echina, meaning spiny, and derma, meaning skin. 1, class Crinoidea, sea lilies and feather stars. 2, class Asteroidea. Sea stars. 3, class Ophiuroidea, basket stars and brittle stars. 4, class Echinoidea. Sea urchins and sand dollars. 5, class Holothuroidea, sea cucumbers, and 6, class Concentricycloidea. Sea daisies, which live on waterlogged wood in the deep sea and were first discovered in 1986.
Why do some genetic conditions skip a generation?
Conditions that are the result of homozygous recessive inheritance can often skip a generation. If a homozygous recessive individual produces offspring with a homozygous dominant individual, then all of their children will be heterozygous and not exhibit the condition. However, should 2 normal-appearing, heterozygous individuals marry, some of their children may have the condition. This also depends on whether the condition is the result of single trait inheritance or the combination of genes and environmental factors. Environmental factors absent in one generation but present in the next. Drought, heat wave, etc., might cause a trait to suddenly reappear.
What is polymerase chain reaction?
Polymerase chain reaction, or PCR, is a laboratory technique that amplifies or copies any piece of DNA very quickly without using cells. The DNA is incubated in a test tube with a special kind of DNA polymerase, a supply of nucleotides, and short pieces of synthetic, single-strand DNA that serve as primers for DNA synthesis. With automation, PCR can make billions of copies of a particular segment of DNA in a few hours. Each cycle of the PCR procedure takes only about 5 minutes. At the end of the cycle, the DNA segment, even one with hundreds of base pairs, has been doubled. A PCR machine repeats the cycle over and over. PCR is much faster than the days it takes to clone a piece of DNA by making a recombinant plasmid and letting it replicate within bacteria. PCR was developed by the biochemist Kary Mullis, 1944. In 1983 at Cetus Corporation, a California biotechnology firm. In 1993 Moelis, along with Michael Smith, 1932, won the Nobel Prize in Chemistry for development of PCR. DNA amplification is a method by which a small piece of DNA is copied thousands of times using PCR. DNA amplification is used in cloning, to detect small amounts of DNA in a sample, and to distinguish different DNA samples, as in DNA fingerprinting.
What is the biological basis for DNA fingerprinting?
DNA fingerprinting, also known as DNA typing or DNA profiling, is based on the unique genetic differences that exist between individuals. Most DNA sequences are identical, but out of 100 base pairs of DNA, two people will generally differ by one base pair. Since there are 3 billion base pairs in human DNA, one individual's DNA will differ from another's by 3 million base pairs. To examine an individual's DNA fingerprint, the DNA sample is cut with a restriction endonuclease, and the fragments are separated by gel electrophoresis. The fragments are then transferred to a nylon membrane, where they are incubated in a solution containing a radioactive DNA probe that is complementary to specific polymorphic sequences.
How is sensory information transmitted to the central nervous system?
Sensory information is transmitted to the central nervous system through a process that includes stimulation, transduction, and transmission. A physical stimulus, example light or sound pressure, is converted into nerve cell electrical activity in a process called transduction. The electrical activity is then transmitted as action potentials to the central nervous system.
What is the name of the bird that perches on the black rhinoceros's back?
The bird, a relative of the starling, is called an oxpecker, a member of the Sternidae family. Found only in Africa, the yellow-billed oxpecker, Buphagus africanus wingspan, is widespread over much of western and central Africa, while the red-billed oxpecker, Buphagus erythraincus wingspan, lives in eastern Africa from the Red Sea to Natal. 7 to 8 in, 17 to 20 centimeters, long with a coffee-brown body. The oxpecker feeds on more than 20 species of ticks that live in the hide of the black rhinoceros. Diceros bicornis, also called the hook-lipped rhino. The bird spends most of its time on the rhinoceros or on other animals, such as the antelope, zebra, giraffe, or buffalo. The bird has even been known to roost on the body of its host. The Relationship between the oxpecker and the rhinoceros is called mutualism. The bird feeds on the rhinoceros's ticks, benefiting both the bird and the rhinoceros. In addition, the oxpecker, having much better eyesight than the nearsighted rhinoceros, alerts its host with its shrill cries and flight when danger approaches.
What was the United States' first national park?
On March 1, 1872, an act of Congress signed by Ulysses S. Grant established Yellowstone National Park as the first national park. The action inspired a worldwide national park movement.
What colored pigments are produced by lichens?
Lichens are often strikingly colored because of pigments that play a role in protecting the photosynthetic partner from the destructive action of the sun's rays. These pigments can be extracted from lichens and used as natural dyes. The traditional method of manufacturing Scotland's famous tweeds makes use of fungal dyes. Synthetic dyes are now commonly used. Orchil, used to dye woolens, is a pigment produced specifically by lichens. The lichen Rosella tincturatoria is used to produce litmus, a widely used acid-base indicator.
What are some beneficial insects?
Beneficial insects include bees, wasps, flies, butterflies, moths, and others that pollinate plants. Many fruits and vegetables depend on insect pollinators for the production of seeds. Insects are an important source of food for birds, fishes, and many animals. In some countries, such insects as termites, caterpillars, ants, and bees are eaten as food by people. Products derived from insects include honey and beeswax, shellac, and silk. Some predators such as mantises, ladybugs, or lady beetles, and lacewings feed on other harmful insects. Other helpful insects are parasites that live on or in the body of harmful insects. For example, some wasps lay their eggs in caterpillars that damage tomato plants.
What is a codon?
A codon is the three-unit sequence, OG, AGC, etc. of mRNA nucleotides that codes for a specific amino acid. Since there are only 20 commonly used amino acids and 64, 4x4x4, possible codon sequences, the genetic code is described as both degenerate and unambiguous. Each codon codes for only one amino acid, but each amino acid may have more than one matching codon.
What is the function of chloroplasts?
Chloroplasts are the structural and functional units where photosynthesis takes place, the process whereby green plants use light energy for the synthesis of organic molecules from carbon dioxide and water, with oxygen released as a byproduct. They contain the green pigment chlorophyll, which traps light energy for photosynthesis.
How do steroid hormones differ from non-steroid hormones?
Steroid hormones such as estrogen and testosterone enter target cells and directly interact with the DNA in the nucleus. Non-steroid hormones such as adrenaline generally do not enter the target cell but instead bind to a receptor protein found on external cell membranes. This then causes a sequence of metabolic effects.
Why was the Darwin-Wallace theory so ridiculed?
The work of Darwin and Wallace generated controversy on at least two fronts. First, their theory directly countered Christian teaching on the immutability of species and their special creation by God. Second, by presenting arguments for common descent. Darwin and Wallace showed that humans were related to other animals, apes in particular. This insulted those who felt that humans were unique and not part of the animal kingdom. However, it should be noted that as the work was disseminated among scientists, it gained general acceptance in the late decades of the 19th century.
What naturally occurring substance provides protection against viral infections?
Interferons protect the adjacent cells against viral penetration. Interferons are glycoproteins produced by body cells upon exposure to a virus. In 1957, Alec Isaacs, 1921–1967, and Jean Lindenman, 1924, identified a group of over 20 substances that were later designated as alpha, beta, and gamma interferons.
What purpose do the gills of mushrooms serve?
Gills, which can be present on the undersurface of a mushroom's cap, serve two main purposes. The first is to maximize the surface area on which spores are produced, allowing a very large number of spores to be produced. The second purpose is to help hold up the cap of the mushroom. Spores are produced in the basidia specialized cells that line the surface of the gills. It has been estimated that a mushroom with a cap that is 3 in 7.5 centimeters in diameter can produce as many as 40 million spores per hour.
How long does it take the average spider to weave a complete web?
The average orb weaver spider takes 30 to 60 minutes to completely spin its web. These species of spiders, order Araneae, use silk to capture their food in a variety of ways, ranging from the simple tripwires used by large bird-eating spiders to the complicated and beautiful webs spun by orb spiders. Some species produce funnel-shaped webs, and other communities of spiders build communal webs. A completed web features several spokes leading from the initial structure. The number and nature of the spokes depend on the species. The spider replaces any damaged threads by gathering up the thread in front of it and producing a new one behind it. The orb web must be replaced every few days because it loses its stickiness and its ability to entrap food.
How do cells become tissues?
Tissues are comprised of different types of cells that have a common purpose or function. Multicellular organisms are so complex that tissues are needed to perform vital functions such as support, transportation, and the movement of animal cells. In animals, the groups of cells that form tissue may become structurally linked via their cytoskeletons and membranes. As an example, epithelial cells are anchored to underlying connective tissue by a basement membrane. These cells are often exposed to mechanical stress, and a system of cell junctions between epithelial cells helps them respond when stretched.
Do all muscle cells work the same way?
Although all muscles work by contracting, not all muscle types have sarcomeres, the muscle contraction units. Cardiac muscle cells have sarcomeres but use different support structures during contraction than those found in skeletal muscles. Smooth muscle cells don't use sarcomeres at all.
How do cells respond to cellular signals?
In order to respond to a signal, a cell needs a receptor molecule that recognizes the signal. A cell's response to a specific signal varies according to the signal. Some signals are local signals, example, growth factors, while others act as distance signals, example hormones. There are two basic types of hormones: those that bind to receptors on the cell surface and those whose receptors are found within cytoplasm. Both types cause the cellular machinery to change its activities.
How do antibiotics differ from antibacterials?
Antibiotics and antibacterials both interfere with the growth and reproduction of bacteria. Antibiotics are medications for humans and animals. Antibacterials, found in soaps, detergents, health and skincare products, and household cleaners. Are used to disinfect surfaces and eliminate potentially harmful bacteria.
How are macromolecules built?
There are 4 types of very large molecules that are important to life. These are carbohydrates, nucleic acids, lipids, and proteins. Although these are quite diverse in terms of structure, size, and function, the same mechanisms build and break them down. All are comprised of single units linked together to create a chain. Similar to a freight train with many cars. All the monomers or single units contain carbon. All monomers are linked together through a process known as dehydration synthesis, which literally means building by removing water. A hydrogen atom, H, is removed from one monomer, and a hydroxide, O, group is removed from the next monomer in line. Atoms on the ends of the 2 monomers will then form a covalent bond to fill their electron shells, thereby building a polymer. All polymers are broken down by the same method, hydrolysis. Hydrolysis means breaking with water. By adding H2O, which contains hydrogen and hydroxide groups, back to the monomers, the bond is broken and the macromolecule separates into smaller pieces.
What are applications of DNA fingerprinting?
DNA fingerprinting is used to determine paternity in forensic crime analysis. In population genetics to analyze variation within populations or ethnic groups, in conservation biology to study the genetic variability of endangered species. To test for the presence of specific pathogens in food sources. To detect genetically modified organisms either within plants or food products, in evolutionary biology to compare DNA extracts from fossils to modern-day counterparts, and in the identification of victims of a disaster.
How do fleas jump so far?
The jumping power of fleas comes both from strong leg muscles and from pads of a rubber-like protein called resilin. The resilin is located above the flea's hind legs. To jump, the flea crouches, squeezing the resilin, and then it relaxes certain muscles. Stored energy from the resilin works like a spring, launching the flea. A flea can jump well both vertically and horizontally. Some species can jump 150 times their own length. To match that record, a human would have to spring over the length of 2.25 football fields or the height of a 100-story building in a single bound. The common flea, Pulex irritans, has been known to jump 13.33 centimeters in length and 7.25 18.4 centimeters in height.
What is spectroscopy?
Spectroscopy includes a range of techniques to study the composition, structure, and bonding of elements and compounds. The different methods of spectroscopy use different wavelengths of the electromagnetic spectrum to study atoms, molecules, ions, and the bonding between them.
What is Lamarckian evolution?
The French biologist Jean-Baptiste de Lamarck, 1744–1829, is credited as the first person to propose a theory that attempts to explain how and why evolutionary change occurs in living organisms. The mechanism Lamarck proposed is known as the inheritance of acquired characteristics, meaning that what individuals experience during their lifetime will be passed along to their offspring as genetic traits. This is sometimes referred to as the theory of use and disuse. A classic example of this would be the giraffe's neck. Lamarckian evolution would predict that as giraffes stretch their necks to reach higher branches on trees, their necks grow longer. As a result, this increase in neck length will be transmitted to egg and sperm such that the offspring of giraffes whose necks have grown will also have long necks. While Lamarck's idea was analytically based on available data, giraffes have long necks and give birth to offspring with long necks as well. He did not know that. In general, environmental factors do not change genetic sequences in such a direct fashion.
Are there differences between prokaryotic and eukaryotic ribosomes?
Prokaryotic and eukaryotic ribosomes resemble each other structurally, but they are not identical. Prokaryotic ribosomes are smaller in size, contain fewer proteins, have smaller RNA molecules, and are sensitive to different inhibitors of protein synthesis.
How do schooling fishes swim so close without colliding?
A fish school travels in a smooth movement. Each fish responds to movements of other fish, such that if one fish changes direction, all of the others will move accordingly. With eyes placed on the side of the head, fishes can see what is next to them and use this to move. A complex combination of hearing, lateral line system, sight, and smell allows fishes to establish position and direction in a school.
What is the largest group of vertebrates?
The largest group of vertebrates is fishes. They are a diverse group and include almost 21 0,000 species more than all other kinds of vertebrates combined. Most members of this group are osteocytes, or bony fishes, which include bases, trout, and salmon.
What is a monkey ball tree?
The Osage orange tree, Maclura pomifera, produces large, green, orange-like fruits. The fruit is roughly spherical, 3.5 to 5 in. 8.8 to 12.7 centimeters, in diameter, and have a coarse, pebbly surface.
What is the Sanger technique?
The Sanger technique was developed by Fred Sanger, 1918, in the 1970s. It is also referred to as shotgun sequencing and is a common method used for DNA sequencing. The basics of this method involve using dideoxynucleotides. Instead of the deoxynucleotides normally found in DNA. Dideoxynucleotides are nucleotides missing a hydroxyl group at the site where one nucleotide usually joins to another nucleotide. If the hydroxyl group is missing, chain elongation stops. The process can be done manually or by automation. The cost of sequencer is approximately $100,000. In the automated process, a short DNA primer is synthesized that is complementary to the template to be analyzed. Electrophoresis is used to separate the bands produced. A different fluorescent label is used on each of the dideoxynucleotides. Adenine, guanine, thymine, cytosine, and these are added to the template. Once hybridization occurs, the chain elongation stops and large numbers of fluorescent bands are produced and read using a laser. The laser then transmits the sequence into nitrogen bases.
What is fish?
Fish, fluorescent in situ hybridization, is a method in which a clone gene is painted with a fluorescent dye and mapped to a chromosome. In fish, cells are arrested in the metaphase stage of mitosis and placed on a slide where they burst open, spreading chromosomes over the surface. A fluorescent labeled DNA probe of specific interest is placed on the slide and incubated long enough for hybridization to occur. The slide is then viewed under a fluorescence microscope that focuses ultraviolet light on the chromosomes. Hybridized regions of the chromosome fluoresce. This is a type of karyotypic analysis that can generate a physical map matching clones and gene markers to specific parts of a chromosome.
Can a cell survive in complete darkness?
Yes, cells can survive in complete darkness. A new theory on the origin of life argues that living systems arose in small compartments of total darkness located within iron sulfide rocks that were formed by hot springs on the seafloor. Cells of plant roots live in total darkness and carry out all normal plant cell activities, with the exclusion of photosynthesis. And, if you think about it, cells located in the very middle of your body also live in a pretty dark environment.
What government departments fund the Human Genome Project?
In the United States, the project is funded primarily by the Department of Energy, DOE, and the National Institutes of Health, NIH. Funding from the DOE grew from a need to research the effects of both radiation and the chemical byproducts of energy production on genes. In Europe, funding was provided by the European Commission, charities, and national research councils.
How are anabolic steroids harmful to those who use them?
Anabolic, protein building. Steroids are drugs that mimic the effects of testosterone and other male sex hormones. They can build muscle tissue, strengthen bone, and speed muscle recovery following exercise or injury. They are sometimes prescribed to treat some types of anemia as well as osteoporosis in postmenopausal women. Anabolic steroids have become a lightning rod of controversy in competitive sports. The drugs are banned from most organized competitions because of the dangers they pose to health and to prevent athletes from gaining an unfair advantage. Adverse effects of anabolic steroids include hypertension, acne, edema, and damage to liver, heart, and adrenal glands. Psychiatric symptoms can include hallucinations, paranoid delusions, and manic episodes. In men, anabolic steroids can cause infertility, impotence, and premature balding. Women can develop masculine characteristics such as excessive hair growth, male pattern balding, disruption of menstruation, and deepening of the voice. Children and adolescents can develop problems in growing bones, leading to short stature.
Who proposed using magnetic resonance imaging for diagnostic purposes?
The concept of using MRI to detect tumors in patients was proposed by Raymond Damadian. 1936, in a 1972 patent application. The fundamental MRI imaging concept used in all present-day MRI instruments was proposed by Paul Lauterbur. 1929, in an article in Nature in 1973. Lauterbur and Peter Mansfield, 1933. Were awarded the Nobel Prize in Physiology or Medicine in 2003 for their discoveries concerning magnetic resonance imaging. The main advantages of MRI are that it not only gives superior images of soft tissues, like organs, but it can also measure dynamic physiological changes in a non-invasive manner, without penetrating the body in any way. A disadvantage of MRI is that it cannot be used for every patient. For example, patients with implants, pacemakers, or cerebral aneurysm clips made of metal cannot be examined using MRI because the machine's magnet could potentially move these objects within the body, causing damage.
Who discovered lysosomes?
Lysosomes are a relatively modern discovery in cell biology. They were observed by Christian de Duva, 1917, in the early 1950s. In 1955, after 6 years of experiments, de Duva was convinced that he had found an organelle that had not been previously described and was involved in intracellular lysis, digestion. He named the organelle a lysosome. This organelle was the first to be described entirely on biochemical criteria. The results were later verified using electron microscopy. In 1974, de Duva, Albert Claude, 1898–1983, and George Pilati, 1912, shared the Nobel Prize in Physiology or Medicine for their work detailing the functions of the lysosome.
Why is DNA important?
DNA is the chemical inside the nucleus of a cell that carries the genetic instructions for making living organisms.
How do viruses enter their host cells to reproduce?
A virus is able to enter a host cell by either tricking the host cell to pull it inside, as the cell would do to a nutrient molecule, or by fusing its viral code with either the host cell wall or membrane and then releasing its genes into the host. Some viruses inject their genetic material into the host cell, leaving their empty viral coats outside of the host cell.
What are demyelinating diseases?
Demyelinating diseases involve damage to the myelin sheath of neurons in either the peripheral or central nervous system. Multiple sclerosis, MS, is a chronic, potentially debilitating disease that affects the myelin sheath of the central nervous system. The illness is probably an autoimmune disease. In MS, the body directs antibodies and white blood cells against proteins in the myelin sheath, surrounding nerves in the brain and spinal cord. This causes inflammation and injury to the myelin sheath. Demyelination is the term used for a loss of myelin, a substance in the white matter that insulates nerve endings. Myelin helps the nerves receive and interpret messages from the brain at maximum speed. When nerve endings lose this substance, they cannot function properly, leading to patches of scarring, or sclerosis. The result may be multiple areas of sclerosis. The damage slows or blocks muscle coordination, visual sensation, and other functions that rely on nerve signals. In the autoimmune disorder known as Guillain-Barré syndrome, the body's immune system attacks part of the peripheral nervous system. The immune system starts to destroy the myelin sheath that surrounds the axons of many peripheral nerves, or even the axons themselves. The myelin sheath surrounding the axons speeds up the transmission of nerve signals and allows the transmission of signals over long distances. In diseases such as Guillain-Barré, in which the peripheral nerves' myelin sheaths are injured or degraded, the nerves cannot transmit signals efficiently. Consequently, muscles begin to lose their ability to respond to the brain's commands. Commands that must be carried through the nerve network. The brain also receives fewer sensory signals from the rest of the body, resulting in an inability to feel textures, heat, pain, and other sensations. Alternately, the brain may receive inappropriate signals that result in tingling, crawling skin, or painful sensations. Because the signals to and from the arms and legs must travel the longest distances, these extremities are most vulnerable to interruption. The first symptoms of this disorder include varying degrees of weakness or tingling sensations in the legs. In many instances, the weakness and abnormal sensations spread to the arms and upper body. In severe cases, the patient may be almost totally paralyzed since the muscles cannot be used at all. In these cases, the disorder is life-threatening, potentially interfering with breathing and, at times, with blood pressure or heart rate, and is considered a medical emergency. Such a patient is often put on a respirator to assist with breathing and is watched closely for problems such as an abnormal heartbeat, infections, blood clots, and high or low blood pressure. Most patients, however, recover from even the most severe cases of Guillain-Barré syndrome, although some continue to have a certain degree of weakness.
Why are fevers dangerous?
Fever is usually an indication of bacterial or viral infection. While there is still debate about whether fevers actually speed up the body's inflammatory response to infection, there appears to be no clinical evidence that reducing fever slows healing. Approximately 4% of children between the ages of 3 months and 5 years experience febrile seizures. It has been commonly thought that high fevers in children could cause denaturation of central nervous proteins, leading to seizures and possibly brain damage. However, the truth of that theory has not been fully determined.
What are succulents?
a group of more than 30 plant families including the amaryllis, lily, and cactus families 182 form what is known as the succulents, from the Latin term succulentus, meaning fleshy or juicy. Most members of the group are resistant to droughts as they are dry weather plants. Even when they live in moist, rainy environments, these plants need very little water.
What is tropism?
Troism is the movement of a plant in response to a stimulus. The categories include chemotropism, a response to chemicals by plants in which uncurling of leaves may occur. Gravitropism, formerly called geotropism, a response to gravity in which the plant moves in relation to gravity. Shoots of a plant are negatively geotropic, growing upward, while roots are positively geotropic, growing downward. Hydrotropism, a response to water or moisture in which roots grow toward the water source. Paraheliotropism, a response by the plant leaves to avoid exposure to the sun. Phototropism, a response to light in which the plant may be positively phototropic, moving toward the light source, or negatively phototropic, moving away from the light source. Main axes of shoots are usually positively phototropic, whereas roots are generally insensitive to light. Thermotropism, a response to temperature by plants. Thigmotropism or haptotropism, a response to touch by the climbing organs of a plant. For example, the plant's tendrils may curl around a support in a spring-like manner.
What are diatoms?
Diatoms are microscopic algae of the phylum Chrysophyta in the kingdom Protista. Almost all diatoms are single-celled algae and dwell in both fresh and salt water. They are abundant in the cold waters of the northern Pacific Ocean and the Antarctic. Diatoms are yellow or brown in color, and are an important food source for marine plankton and many small animals. Diatoms have hard cell walls. These shells are made from silica that has been extracted from the water. It is unclear how the extraction of silica from water is accomplished. When they die, their glassy shells called frustules sink to the bottom of the sea and harden into rock called diatomite. One of the most famous and accessible diatomites is the Monterey Formation along the coast of Central and Southern California.
What is post-translational processing?
The process of translation uses an mRNA pattern to produce a string of amino acids, technically known as a polypeptide. To turn a polypeptide into a protein with the shape that is crucial to its function requires several more steps. Sugars, lipids, phosphate groups, or other molecules may be added to the amino acids in the chain. The polypeptide may be trimmed as well, the first few amino acids may be removed. Some proteins, like insulin, only become functional after cleavage by an enzyme. Because of this ability to create variations from the basic polypeptide chain. A given gene may code for several different functional structures. This helps to explain the unexpectedly small size of the human genome. Our 35,000 or so genes actually code for several 100,000 different proteins.
What criteria are used to identify emerging infectious diseases?
Emerging infectious diseases are ones that are new, changing, have shown an increase in incidence, or have the potential to increase in the near future. There are several criteria used to identify an emerging infectious disease, including 1. A disease causing symptoms that are clearly different from all other diseases. 2. Diagnostic techniques that allow the identification of a new pathogen. 3. A locally defined disease that becomes widespread. 4. A rare disease that increases in occurrence and becomes common. 5. A mild disease that becomes severe. 6. A slowly developing disease that increases in lifespan.
How much cholesterol is needed in your diet?
None. The body is capable of making all the cholesterol it needs. Almost all cells can synthesize some cholesterol, but most require more to maintain plasma membranes. This additional cholesterol, which is found in the body, is synthesized by the liver. All cell membranes contain cholesterol, and it is also used in producing bile salts and steroid hormones.
Are any bacteria visible to the naked eye?
Epilopiscium fischelsoni, which lives in the gut of the brown surgeonfish. Acanthurus nigrofuscus, is visible to the naked eye. It was first identified in 1985 and mistakenly classified as a protozoan. Later studies analyzed the organism's genetic material and proved it to be a bacterium of unprecedented size. 0.015 in, 0.38 millimeters, in diameter, or about the size of a period in a small print book.
What is ethology?
Ethology emerged in the mid-1930s and was first recognized as a subdiscipline of biology in Europe. Ethology differed from traditional biological studies of animals in that scientific principles were applied to the study of animal behavior, with practitioners using both field observations and laboratory experiments. The field was developed and first recognized as a science in Europe. where experimental conditions were kept as natural as possible.
What is wood?
Wood is the accumulated secondary xylem of a plant. Generally, the wood used commercially is from plant stems rather than plant roots. Wood located near the center of a tree trunk is called hardwood. Its cells are infiltrated with gums and resins from the aging secondary xylem. Hardwood is often darker in color than wood nearer to the vascular cambium. Wood closer to the vascular cambium is called sapwood. It is actively involved in transporting water within the plant.
Is IQ genetically controlled?
This is actually two questions. First, can intelligence be measured quantitatively? And second, is there a correlation between intelligence and certain genotypes? Both questions can be answered in the negative. As our definition of intelligence has evolved, it has become more difficult to assign a single number to the trait of intelligence. Therefore, it is almost impossible to demonstrate which gene sequences correlate to high IQ values. Even though our understanding of the genome is broadening, so too is our definition of intelligence. It becomes less and less likely that we will be able to find one or two genes that determine all of the facets of intelligence. Considering the importance of environment in determining phenotype, IQ is most likely a multifactorial trait.